Hello guys, hope that you're good. Today I will tell you about PAMPs or pathogen associated molecular patterns which are present on the surface of different kind of bacteria, viruses, parasites, fungi, etc. and are recognized by cells of our immune system in order to generate responses for the clearance of these pathogens from our bodies. So if you look at immune responses in our bodies, these responses are generated by two types of cells. Cells of the innate immune system and cells of the adaptive immune system. So cells of the innate immune system, which include these dendritic cells, macrophages, the mast cells, natural killer cells, granulocytes, just like the neutrophils, etc., these cells recognize and respond, to different kind of pathogens, and how they recognize the pathogens. This innate immune system or cells of the innate immune system recognize antigens common in a group of pathogens called PAMPs or pathogen-associated molecular patterns. It means that these cells have receptors for the recognition of groups of pathogens. It's like groups. Gram-positive bacteria is a group of bacteria. Gram-negative bacteria is another group of bacteria. Similarly, viruses, different groups of viruses, or different parasites or fungi are recognized, using common antigens which are present on the surfaces of these bacteria. So this system would recognize all gram-positive bacteria in the same way and would respond in the same way, by just engulfing them, doing phagocytosis, or by just, uh, secreting different kinds of chemokines and cytokines to generate these inflammatory responses inside our bodies. So for a group of pathogens with common antigens on their surfaces, all these cells would respond in the same way. And we also know about other attributes of the innate immune system that we already know that it's not very specific, because if you are just responding to common antigens on the surfaces of groups of bacteria, viruses, or parasites, then the responses generated would not be specific against a particular pathogen that has entered your body. These responses actually are against all, pathogens in that particular group. So these responses usually would lead to generation of other responses, which are known as responses of the adaptive immune system. The adaptive immune system has two branches, humoral and the cell-mediated responses. The humoral responses are generated by the B cells, and cell-mediated responses are generated by the T cells. So the cytotoxic T cells, they can either directly go and kill the cells that are infected, or these cells can also take the help of antibodies and kill the infected cells. So these both of these cells, the T cells, the helper T cells, and the B cells, they are going to recognize highly specific antigens present on the surfaces of different pathogens. For instance, this particular system on the right side, the adaptive immune system, can differentiate among different bacteria. It can specifically kill a bacteria. For instance, it can tell which bacteria is, Salmonella typhi, for instance. But this system cannot tell, on the left side, the innate immune system cannot tell that which bacteria is Salmonella typhi. This system would respond, in a more, general way, while the adaptive immune system would respond in a more specific way, targeting a specific bacteria, which is Salmonella typhi. So this innate immune system would respond the same way in case of all gram-negative bacteria. For instance, as I told you about Salmonella typhi, uh, if it is Pseudomonas, etc., which are all gram-negative bacteria, this system would respond the same way because it recognizes particular PAMPs or patterns on the surfaces of all these, gram-negative bacteria. But this system, the adaptive immune system, would generate specific responses by recognizing, uh, you know, Pseudomonas from Salmonella type. Antibodies generated for Pseudomonas would be different from antibodies that are generated in the case of, uh, you know, Salmonella type. And it is because of the specific antigens that are recognized by this system uh, with the help of these T helper cells, the B cells will produce antibodies against those particular antigens. And those would be specific to these two different kinds of bacterial species. So this one is more specific, and on the left, the innate immune system is considered less specific due to recognition of this particular, pattern or the pathogen-associated molecular patterns. Now, what are the common PAMPs on different pathogens? If you look at the viruses, the glycoproteins on the surface of viruses, these are PAMPs, which, all the viruses which would have these glycoproteins on their surfaces would trigger these innate immune responses, or they would be recognized by cells of the innate immune responses in the same way, and the response of the cells would also be the same. Similarly, DNA, which is present inside the viruses, or RNA, which is present inside the viruses, are also PAMPs of viral origin. So the system, the innate immune system, would respond the same way in the case of this DNA or RNA of viruses. So groups of viruses would have these things in common. These glycoproteins present on the surfaces will be similar, so the system would respond the same way. Similar is the case with gram-positive bacteria. For instance, on gram-positive bacteria, you can find a number of PAMPs, like the DNA, which is present inside these bacteria, is a PAMP. Similarly, the lipopolysaccharide, which are present on the surface of these walls, also are PAMPs. Similarly, the proteoglycans present on the surface are also PAMPs. LTA is also a PAMP. In the case of gram-negative bacteria, you can see that again, DNA, furan, proteoglycans, lipopolysaccharides. This is lipoprotein in the case of gram-positive bacteria. In gram-negative bacteria, you can find these lipopolysaccharides. So lipopolysaccharide and flagellin, in addition, all these are pathogen-associated molecular patterns, and they can be recognized by the different cells of the innate immune system, as I told you. Similarly, in the case of fungi, zymosin, mannan, beta-glycan, GPI anchors the DNA of the protozoa. In this case, all these are, pathogen-associated molecular patterns. Here is a colored sketch, so you can also take help from this particular sketch. How are these PAMPs recognized by cells of the innate immune system? Uh, the PAMPs are recognized with the help of pathogen, recognition receptors, and we call them PRRs. You know, For instance, the PAMPs on the viruses, as I told you, are these glycoproteins or the DNA and RNA. So what are the recognition receptors on the surfaces of the immune cells? Most of them are the toll-like receptors. So the glycoprotein, for instance, of viruses is recognized by these toll-like receptor 2 and toll-like receptor 4. Similarly, the DNA of this virus can be recognized by this toll-like receptor 9. Similarly, RNA of this particular virus can be recognized by other receptors present on the surfaces of cells, which are the toll-like receptor 3, 7, or 8. Similar is the case with gram-positive bacteria. Different elements, different PAMPs like the DNA, lipoprotein, proteoglycans, or LTA can be recognized by these different toll-like receptors is given here. Similar is the case with gram-negative bacteria. So all the PAMPs associated with gram-negative bacteria, different gram-negative bacteria, can be recognized by different toll-like receptors either present on the surfaces of the cells or present on endosomes. Similar is the case with other pathogens, fungi, and protozoa. Different toll-like receptors are involved in this particular process. Okay? And these are not only the toll-like receptors, there are other receptors there as well, which are involved in this uh, process of recognition of the pathogens. Toll-like receptors is one of the biggest group of such receptors which are recognizing different PAMPs, and these receptors are present on different immune cells. Similarly, cytoplasmic node-like receptors are the NLRs are also uh, uh, PRR, like the pathogen recognition receptors. Intracellular retinoic acid inducible gene 1-like receptors are RLR. As you see here, these receptors are also, PRRs. Transmembrane C-type lectin receptors present on the surfaces of cells are also, PRRs. They also are recognizing different patterns. And absent in melanoma, 2-like receptor AIM2 are NODs, nucleotide binding oligomerization domain, or RIG1, retinoic acid inducible G1. These are all different types of, PAMPs, recognition receptors, or pathogen recognition receptors on the surfaces of the immune cells that I have shown you in the very first slide. On the surfaces of these different types of phagocytes, etc., these receptors are present. So where to find these pathogen recognition receptors, or the PAMPs recognition receptors, you can also call it like that. There are 3 different locations inside the cells or outside the cells where you can find these different types of, receptors. These receptors are either present on the surface of the cells, for instance, a TLR5, or TLR4, or TLR2, toll-like receptor 2, 4, 5, 6, etc. These receptors are present on the surface of the cell. They are part of the cytoplasmic membrane, so they can recognize different PAMPs or different pathogens right here at the surface of the cell. Some of the pathogen recognition receptors are present in the endosomes. These include TLR9, or TLR3, or TLR8. So these are other, toll-like receptors which are present on the endosomes. Whereas the endosomes, when the pathogen is phagocytosed and the membrane is, surrounded, around that particular pathogen, that structure is an endosome, phagosome. So inside that phagosome, you can find these different toll-like receptors for the recognition of that particular pathogen. Similarly, uh, apart from the cytoplasmic membrane, you can find uh, these PRRs in the endosomes, and some of the PRRs are present in the cytoplasm. What are the PRRs present in cytoplasm? These include the nod-like 2 receptors and the RIG1 receptor, as I told you about it in the previous slide. So these nod-like receptors, these nucleotide binding oligomerization domain, and RIG1, the retinoic acid inducible gene 1, these are PRRs which are present in the cytoplasm. And some of the PRRs are secreted, into tissues, just like into blood or other body fluids. You can find some of the PRRs, just like the mannan-binding protein, which is secreted by cells of liver, the hepatocytes, or the C-reactive protein, you have heard about it, the surfactant protein A, SPA, surfactant protein D, SPD, or collectin liver 1, CL1, and phycolins. All these are secretory, pathogen recognition receptors. So how do these PRRs recognize pathogens? These PRRs can recognize specific pathogens. They can bind to them, and they can then come and bind to, another domain can bind to specific receptors on the surface of the cells, and these cells would generate immune responses in different ways. Sometimes I will tell you about these responses in the next slide. So what are the cells that have all these different types of the pathogen recognition receptors? These cells include macrophages, dendritic cells, mast cells, endothelial cells, mucosal epithelial cells, lymphocytes, and fibroblasts. So all these cells have these different types of pathogen recognition receptors, either on their membranes or as part of the endosome, are free in the cytoplasm, are secreted into tissues just like the blood and other body fluids. This is the functional classification of uh, the pathogen recognition receptor. It can be classified into two different classes: endocytic pattern recognition receptors and the signaling pattern recognition receptors. So what are the endocytic pattern recognition receptors or phagocytic receptors? These are present on the surface of phagocytes and promote attachment of microorganisms to phagocytes, leading to their subsequent engulfment and destruction. And there are different, types of them. Some are called the mannose receptors, dectin-1 scavenger receptors, opsonin receptors, N-formyl methionine receptors. These are 5 different types of the endocytic pattern recognition receptors, which are doing this particular job: attachment of microorganisms to phagocytes, and then leading to their subsequent engulfing and destruction. So these are present on the surfaces of these phagocytes, and these are important with respect to attachment of these microbes, these bacteria, viruses, etc., and that leads to their phagocytosis and then destruction inside the cells. The second types are the signaling pattern recognition receptors. So what is their job? Their job is binding up microbial PAMPs to signaling pathogen recognition receptor, which promotes the production of what? Number one, inflammatory cytokines such as interleukin 1, tumor necrosis factor alpha, and interleukin 12. Antiviral cytokines called type 1 interferons such as interferon alpha and interferon beta. And number 3, Chemotactic factors such as chemokines, interleukin-8, MCP1, and RANTES, etc. Number 4, antimicrobial peptides such as human defensins and cathelicidins. These molecules are crucial to initiating innate immunity in adaptive immunity. So these signaling pattern recognition receptors again are of 4 different types. This is the job that these signaling pattern recognition would do once they are bound to these PAMPs on the surfaces of pathogens. What is what are these 4 different types of the signaling pattern recognition receptors? These are number 1, signaling PRRs found on cell surfaces. Just like the toll-like receptor 2, 4, 5, and 6 are present on the surfaces, on the surface of the cell, as I showed you here. These are the signaling PRRs found on the surface of the cell. Some signaling PRRs are found in the membrane of endosomes or phagolysosomes. They are used to degrade pathogens. TLR 3, 7, 8, and 9. These are present here. TLR 3, 7, 8, and 9. These are present on the surfaces of these lysosomes or phagolysosomes or endosomes. Some of these signaling pattern recognition receptors, uh, uh, these signaling PRRs and DRRs are found in the cytoplasm. And I told you about these, which are present in the cytoplasm. These include the node in RIG1, as you can see here. And the 4th type are the secreted signaling PRRs, which are secreted outside the cell, and these PRRs then join different body fluids, just like the blood. You can find them inside the blood. And what is their job? They activate the complement system and different kind of phagocytes. And what are these secretory PRRs? These include the mannan-binding protein, for instance, C-reactive protein, surfactant A, surfactant protein D, collectin liver 1, and ficolytic. So these are secreted outside the cell, and then they are involved in activation of the complement system and different kind of phagocytes. Different, if you look at the functional classification, we had 2 major classes again for the endocytic pattern recognition receptor or phagocytic receptors. We have 5 subclassifications. And for the signaling pattern recognition receptors, we have 4 classifications, which is majorly based on the location of these receptors, which can either be outside the cell or inside the cell. So that was all about the pathogen recognition receptors. The importance of these pathogen recognition receptors is that pathogens that are recognized in groups would you can expect that the immune responses generated against such kind of pathogens would not be, in most cases, be strong enough to get rid of such pathogens. But these, as you saw, that some of these responses would lead to adaptive immune responses, and adaptive immune responses would generate a strong defense against the pathogens, and we would get rid of the pathogens as a result of that. So if you have any questions, you can just leave a comment, and for more videos you can watch my channel, GDV Education. Thank you very much for watching.